All right. So module 5.5 is photosynthesis. And by the way, you may see if you look at the OCRA specification that this order isn't exactly chronological with the spec, but I've done that because it makes sense to teach certain modules before other modules. It's just better for understanding, in my opinion. So I've laid it out, but I will try and make sure that we go through the bulk of everything, okay? So, photosynthesis, this module is better than what you think. It's better than probably what you remember, if you've already been taught this, because there's quite a lot of, monotonous, tedious explanations and diagrams. But I promise that I'll try to make this as clear as possible and concise as possible. So here we've got a leaf, and if you zoom in on the cross section of a leaf, this is what you will see. Okay, so, I mean, this diagram shouldn't be anything new to you. This diagram shows the different layers of a leaf, and specifically, we want to be looking at the palisade mesophyll layer. This contains palisade mesophyll cells, or no, sorry, you just say palisade cells, and the palisade cells are full of chloroplasts. Now, chloroplasts are the site of photosynthesis. Once again, this is all GCSE knowledge. But if I actually zoom in on a chloroplast, it was very hard to draw it on this diagram, but if I actually zoom in on a chloroplast, this is what I will see, okay? So, once again, it's lagging a bit. But so chloroplasts are, this is a chloroplast here. And what do we have? Well, we have we have a very distinctive double membrane. And that double membrane, if you can remember from the first module in the year 12 course, a double membrane is called an envelope. Okay, so the chloroplast has a double membrane called the chloroplast envelope, and and stored within that inner membrane of the envelope is basically the stroma. So the stroma is the fluid. It's pretty. I like to think of it as being very similar to the cytoplasm because it's mostly water. It's full of ions, and it's the site of many chemical reactions. So I mean, it's sort of like the cytoplasm of an individual chloroplast as opposed to like the cytoplasm of the whole cell. This is like a little personal chloroplast for the a personal cytoplasm for the chloroplast. Okay, now within that stroma we have many things. Okay, the stroma is the site of the light independent reaction. By the way, we'll get onto this more later. But we have these stacks of membrane. We call the membrane thylakoid membrane. That is the specific variety of membrane that these stacks are made out of. And we called one stack a granum. Now, grana is the plural, and grana are the site of the light dependent reaction. Um, Grana are connected by more pieces of membrane. You could think of these like pieces of rope called intergranal lamellae. Okay, so so there's also circular DNA loops in the cytoplasm. Sorry, in the stroma, that you don't really need to know anything more about these than the fact that they store the chloroplast's DNA, and that they end up being transcribed. That's all you really need to know. But I want you to look now at the grana. Specifically, we'll look at this granum here, and I want you to imagine that we zoom in very, very close on the granum as far as you can possibly go. Okay, we'll keep zooming in, we'll keep zooming in, we'll keep zooming in, right to that bit there, we'll keep zooming in. Oh my gosh, we've literally gone to the atomic level. The program literally got messed up there. But what you would see if you zoomed in that far at the atomic level would be this. Okay, you would see this diagram. If you zoomed in on a piece of thylakoid membrane, you would see this diagram here. And this diagram, it haunts students when they see it. It is very intimidating. There's a lot going on here, but this is effectively, let me just zoom in to get everything here. This whole diagram is basically the whole of the light dependent reaction, and we call this non-cyclic photophosphorylation. The reason why we call it this is, well, photo means light. It's the light dependent reaction. It requires light, as you can see up here, And we say it's photophosphorylation because the end product is, I guess you could say, indirectly, we're producing ATP. We're phosphorylating ADP to form ATP. So I'll give you a quick run through of everything in this diagram. Okay, so here, so these purple lines represent the thylakoid membrane. Okay, it's the membrane of a granum, and we've zoomed in up on an individual membrane that's basically folded into the granum, and you can see, like, you could think of this like a jumper. You've got your jumper, and you've got loads of little buttons on the jumper. Each rectangle or each structure in this diagram represents a protein, and each protein has its own function. So PS2 and PS1 are both a type of photosystem. The naming is very, very confusing here. It's a very common mistake. People confuse PS2 with PS1 because PS2 comes before PS1 in the process. But for some, I guess the reason why PS2 is named PS2, even though it comes before PS1, is basically because PS2 was discovered after PS1. Okay, but basically, what photosystems are is they are proteins, and they have they have these pigments, these coloured pigments attached to them, um, sort of like chicken pox. You could think of it like someone who's got chicken pox. The pox are like the pigments, and the person is the protein, and they're just full of these pigments. And what those pigments do is, those pigments are what we basically form the definition of what of the different, the pigments have chlorophyll in them. Chlorophyll is a very general term, but it's basically referring to pigments that are strapped to photosystems that allow light to be absorbed. And we've also, I'll get more onto that in a second. But ultimately, what photosystems will do is they will release an electron that will be transported along these electron carriers, which I've shown here. I've shown them as like these smaller proteins, with the red boxes, and basically when the electron moves along there, it it powers these proton pumps that actively pump hydrogen ions, which we also call as protons. They actively transport or actively pump those protons into this thylakoid space. So I've tried to show that here. It's very small up here, but basically you can think of it like you've got your outer thylakoid membrane, you've got your inner thylakoid membrane, or not even outer and inner, just the space stored within that membrane, because it's like a massive sack if you remember from module 2.1. Okay, so basically, yeah, so the electrons move along there, power those proton pumps. The protons get pumped into the thylakoid space, um, and then the same thing happens here because you've got electrons moving along this massive chain, and basically they end up at PS1. PS1 is like, I think of it like a traffic jam. All the electrons can't move along this chain until an electron is released specifically from PS1, and then uh, the electrons here basically move uh, along electron carriers. The same thing happens, producing, actively pumping hydrogen ions into the thylakoid space. But ultimately, the PS1 electron forms a molecule called NADPH, which is a coenzyme. And finally, the hydrogen ions diffuse down a molecule called ATP synthase. But we'll go into this in detail now. Okay, I've tried to give you a brief introduction, a brief explanation of what everything in this diagram represents. It's all right if you don't understand everything I've just said, because it probably was a bit confusing going through everything at once, but I want you to have a general idea of what everything represents. This is called ATP synthase. It's the enzyme that synthesizes ATP. But we'll go into the individual stages now, okay? And any one of these stages, I've seen, this first stage comes up a lot as a 5 marker, okay? So in the in the um, light-dependent reaction, the first thing that happens is a photon is absorbed by PS2. Okay, PS2, it's a photosystem and it can absorb light around the wavelength range of about 680 nanometers. That this is like the how we abbreviate that on the diagram. We write P680, but that means it can absorb light around the 680 nanometer range. Okay, so what happens when PS2 absorbs it? So as I've shown, you've got your so on here you've got your photosystem, and on the diagram I've drawn the protein in yellow. The protein obviously has its pigments attached to it, and there are two main types of pigments. You've got accessory pigments, which are these. I've shown them as orange, okay. And what the accessory pigments do is they are um, that they surround primary pigments, which I've shown in blue, and the accessory pigments that some people might say they're not as important as primary pigments, but what the accessory pigments do is they basically allow a a larger wavelength range of light to be absorbed by the plant, because these primary pigments um, by themselves can't absorb much of a large range of light at all, a large wavelength range. So you need the accessory pigments to come in and basically be like the sidekick. You can think of it like the main character, and then you've got its sidekick. These guys are the sidekick, and what they do is they absorb a photon, as I've shown up here. They absorb that photon, and the I've tried to show this with a little pink arrow here. I'll zoom in on that. That little pink arrow there is the light being absorbed by the accessory pigments, and what happens is we find that electrons become excited. Okay, the electrons from these pigments become excited, but then so they jump up to a really high energy level. Okay, but then they realize they don't have anywhere to put the energy. They can't do anything with that energy, so they're on this really high energy level, sort of like taking an energy drink at like 1 in the morning. You have all this energy, but you can't do anything with it. So the energy, it basically just gets it's not used by the accessory pigments. So what they have to do is they have to pass that energy, the photon, they have to pass it back to neighbouring accessory pigments. Accessory pigments include chlorophyll B. That is a common one that you do need to know of. But ultimately, what happens is this photon gets passed to neighbouring accessory pigments until eventually it reaches the primary pigments at the centre. The primary pigment that you should probably be aware of is chlorophyll A. There are more like a bit like carotene, and there's I mean, if you go on questions, it always comes up as a one marker, but you don't really need to know like every single one. It's not necessary in my opinion. Um, and ultimately, so now the primary pigment has absorbed that photon, but the primary pigment, he's special, okay? The reason why he's special is because he can actually do something with the photon energy. When the PS2 electron absorbs the energy, it gets excited like up here, but the electron can actually be released out of the photosystem. So what we find that this electron gets launched out of the PS2 system, and it bounces along the electron transport chain through electron carriers, which were these proteins that I described earlier. But I want you to think now, this guy has just lost an electron, okay? So he's going to be slightly positively charged, or he will in fact be positively charged. So what we need is, we need to replace those electrons, okay? After stage one has occurred, where the photon's been absorbed and the PS2 electron has been released. What we need now is we need to somehow replace those electrons. So where does that come from? It comes from, and I think I should probably draw this in like blue. But ultimately, what happens is water gets split up, okay? And if I go over here, we call this photolysis of water. Water becomes photolysed, okay? And so water it absorbs light and it splits into 2H plus at half O2 and 2 electrons. Okay, the electrons that are released from this reaction, this electron pair, replaces the PS2 electrons. Okay, so if we go back up here, the electrons here that are released replace the PS2 electrons, okay, and then as the electrons continue, as the original PS2 electrons continue to move across the chain, because you've got to remember there's millions of these, there's not just one electron carrier, there are millions. So basically, you end up with a chain of electrons following each other because as those PS2, as those water molecules electrons move along the transport chain, then you'll have to have more electrons replace them and more electrons replace them, and you've got multiple of these being excited at once. So it's a massive chain. You end up with this humongous electron chain. It's like I'm trying to give you a good analogy here. Um, like on like a playground. Imagine there's a kid like everyone's playing tig, um, but there's like five people are the chaser, and then like everyone's running for everywhere at once. Like there's not just one place where the electrons are coming from. There's multiple people at once chasing everyone. So there's multiple sources of electrons moving along that transport chain. Because there's multiple chasers that are pushing everyone in that direction. You know what I mean? That analogy wasn't the best, but it's pretty. I hope the previous explanation has been sufficient for you to get this concept. Okay, so as the electrons, specifically PS2 in this context, because they come from PS2, as the PS2 electrons move along the electron transport chain, they provide the energy to actively pump um, protons or hydrogen ions, H plus ions, that are present in the stroma across the thylakoid membrane into the thylakoid space. Okay, so I described that earlier. The energy from here, it powers these proton pumps to actively transport H plus ions into this space up here. Okay, now because there are so many hydrogen ions up here, because they are so packed in one tiny space, and there's relatively few down here, what we establish is called an electrochemical gradient. So I've shown that here. So it's an electrochemical gradient, and the reason why it is called an electrochemical gradient and not just a chemical gradient is because you've got two things at play here. Not only do you have the hydrogen ion particles bumping into each other, banging and clashing and colliding into each other because there's so many in such a small place, like marbles, if you shook them around in a container, you've got that factor which is a chemical gradient causing them to bang into each other and repel, but you've also got an electrical gradient. And the reason why it's an electrical gradient is because, as you can see, they're both positively charged. They're both positively charged, and therefore they repel each other. They repel each other. So they want to get as far away as possible, not just because they're banging into each other as particles, but because their charges are also repelling. So you've got those two factors at play here. So now you've established an electrochemical gradient, and what happens is the hydrogen ions diffuse back down their electrochemical gradient by through an enzyme called ATP synthase. Now, this guy is special in stage 3. ATP synthase is special because when hydrogen ions diffuse across it, what this guy can actually do is have you ever been to like an arcade and there's them machines where you put in a pound coin and then you turn the cog and it like throws out a little toy back? It gives you like a little toy, like in an egg plastic cup. That like or like anything really, like where you turn the cog and you get like a little toy back, come back out. This is like ATP synthase. The H plus ions have been pushed through it, and it turns a cog that causes ADP and PI to be grabbed by ATP synthase in its active site, and then it overcomes the activation energy to allow ATP to form. Okay, I think that's pretty phenomenal. It's pretty mad how how plants have evolved to have those present in them is honestly phenomenal. But anyway, so yeah, that produces ATP, and the hydrogen ions were back where they started in the stroma. Okay. Um, I want you to, but for stage four, I want to bring your attention to basically to up here where we have PS1, because stage four is the last stage of this, and it includes the photosystem one. Um, the electrons in the electron carrier in the electron transport chain have basically all been built up at PS1, and the transport chain cannot continue until a PS1 electron is released, because the PS2, I want to bring your attention to the fact that the PS2 electrons have all built up here, but like a traffic jam, they're waiting for PS1 electrons to be released so that the final stage can occur. Okay, so the photon is absorbed by PS1. PS1 can absorb light around the 700 nanometer range, but ultimately what happens is you've got a very similar thing going on up in PS1 as what occurred down here. Okay, but this time we release PS1 electron pairs out of this photosystem, and the PS1 electron pairs are very special. Okay, the reason being is because when they get zipped along the electron transport chain, they release energy, and some of that energy, is to actively pump hydrogen ions across more of these guys. I just couldn't show them all on the diagram, but some of the energy released by the electron is used. Okay, and this is where it's you have to be very specific here. Some of the PS1 electron energy is used to react NADP with H plus to form NADPH. Okay, that hydrogen ion comes from the stroma. The NADP is a coenzyme. It's present in the stroma as well. But ultimately, this reaction cannot occur with PS2 electrons. Okay, it can't occur with PS2 electrons. So I'm going to actually put a little I'm going to put not PS1. Actually, you won't be able to see that if I do it in that color. So let me just undo that. I'm going to put, if it will let me, for some reason it's lagging, not PS2. This is not PS2. It has to be PS1. So the PS1 electron, he's like in a relay race, the last baton carrier. He's the last dude in the race to hold that baton. And what he does is he gets that electron, or the electron from the PS1 is the last baton holder. And what he does is he reacts with NADP and H plus to form NADPH. Please remember to say that the electron that forms NADPH must be PS1, because the energy of PS1 it's high enough to react with NADP and H plus. PS2, if you remember, he's only got energy coming from light. Okay, but PS1, he's got light energy as well as energy from the other electrons, basically transferring to it, like as they get soaked up, as the electrons here get soaked up by PS1, it increases its energy level, and it's got additional energy from light absorption. Basically, he has a high enough energy to react. Okay, so that is the light dependent reaction. NADPH and NADP are both coenzymes, and this stuff, specifically with like PS2 and PS1, it's very confusing, but you do need to learn it as I've taught you it. The the Na so I'll get on to, actually I'm going to move on to explaining the next part of this before I explain that. So this process was called non-cyclic photophosphorylation. Once again, it absorbs light. Phosphorylation, it produces ATP by phosphorylating ADP, and this is the light dependent reaction. The reason why this is non-cyclic is due to the fact that the electron, okay, it is removed from the electron transport chain. The electron, it starts off here, and it starts off in the thylakoid membrane, and it ends in a molecule. So it doesn't finish where it started. It is removed from the electron transport chain. Now, there's another type of photophosphorylation that you must know called cyclic photophosphorylation. And oh my goodness, if it couldn't have got any more confusing, this photophosphorylation only, and I want to emphasize only, it only works with PS1, okay? So cyclic photophosphorylation is where the no NADPH is made, okay? And you can kind of suss that out by the fact that the the electron isn't released the from the electron transport chain because it is sent back to where it started, okay? The PS1 uh, photosystem absorbs light, absorbs photons, and basically it releases a PS1 electron, which powers the proton pumps to actively pump H plus ions to form the electrochemical gradient. The H plus ions diffuse across ATP synthase. So you've got ATP production going on here, but those electrons are sent straight back. The electrons are sent straight back to PS1. So this process is cyclical, okay? This process, okay, is, I guess, it produces ATP as well as respiration. So and so does non-cyclic as well. ATP production doesn't just come through like respiration; it also comes through this process through photophosphorylation, whether it is cyclic or non-cyclic. So that is important to remember. And once again, it is only PS1 electrons here. Um, it, I don't know. Like, I mean, I guess you just got to learn it that way. I couldn't tell you the intuitive reasoning behind why. But ultimately, a very common question that you want to be aware of is, or at least I've seen this come up multiple times, the explanation of whether PS1 is more important than PS2. Okay, so if we look at our photo systems, which one of these two is the more important uh, team player? Who's the more important guy? So I want you to think about something. If I have a weed killer and it destroys PS2, well, I mean, straight off the bat, non-cyclic photophosphorylation, this whole process, it won't be able to occur because the PS2 electrons have to come from PS2, they have to build up here, they have to provide the extra energy for PS1 electrons. So none of this will occur if this guy is destroyed. But if PS1 is destroyed, so if PS2 is destroyed, and PS1 is still intact, well, you still will be able to, you will still have cyclic photophosphorylation going on here because PS1 are still healthy, they're still functioning, okay? So I guess for that reason, if you actually destroyed PS1, well, non-cyclic photophosphorylation won't occur. None of this will occur because the electrons from PS2 won't have anything to basically carry on the chain. You know, PS1 electrons won't be able to be released to react with NADP and H+. So none of this will occur, okay? But not only will non-cyclic photophosphorylation stop occurring, cyclic photophosphorylation will stop occurring as well, okay? So you're not even going to have ATP production through cyclic photophosphorylation if PS1 gets destroyed, and it is for that reason that this PS1 photosystem is much more important. At least in examiners' eyes, it's much more important than PS2. PS2 with PS1, you can still produce some ATP through cyclic photophosphorylation. Okay. Now the NADPH. Oh my gosh, I forgot to mention this. The movement of the electrons along the electron carrier chain. So basically, you could look at it like the electrons bouncing along here, the electrons bouncing along here to provide ATP. To provide the proton pumps energy, so ATP synthase can produce ATP via the electrochemical gradient. Whew. That entire process is called chemiosmosis. Okay, that was just a little side point. Um, but yeah, so this process, the ultimate goal of the light-dependent reaction through non-cyclic photophosphorylation was to make NADPH, and the NADPH, which we have now made, goes on to react in the Calvin cycle, which is the light independent reaction, and that will occur in the stroma of the chloroplast. Okay, so hopefully that's been useful. Yeah,